So the problem we're concerned with is brain aging. This is typically exhibited by cognitive dysfunction, impaired proteostasis, metabolic issues, and ultimately neurodegeneration and inflammation. The interesting thing about brain aging versus other tissues is that in basically all mammals, some individuals remain cognitively intact to quite advanced ages, while others show gradual decline with aging. In the case of humans, there's kind of two modes of aging-related decline. There's, I guess, what one might call normal aging-related memory loss, which is a gradual decrease in cognitive function over time. And then there's Alzheimer's disease or dementias, where there's a precipitous decline in cognitive function. And so in both of these modes of aggregation or of cognitive decline, there is protein aggregation, which we'll get into in a minute. But just as a little aside for those that don't really or haven't witnessed the disaster that it is dementia, this is just kind of an illustration of cognitive decline in a particular individual. Some artwork by William Uttermohlen, who was, let's see, get this pointer going, who was a somewhat famous artist. Where's the, okay, well, anyhow. Who painted a number of, sorry, perfect, thank you. Who painted a number of self-portraits. So in the upper left is a self-portrait that he painted while cognitively intact. And in 1995, he was diagnosed with Alzheimer's disease and painted a series of self-portraits as the disease progressed. And as you can see, initially he had, well, perhaps an angry demeanor about the whole thing. He had, fine grasp of his artistic talents, but as he progressed in the disease, his use of color and perspective and eventually even paint fell away until in late 2000 he was no longer able to paint at all. So this is just kind of one illustration of the cognitive robbing capacity of dementia. So let's get back to molecules in cells. That's kind of where most of the rest of this is going to go. In Alzheimer's disease and in aging, there are two pathologies in the brain that are prominent and thought to be involved in cognitive decline. That is plaques made of the beta amyloid peptide that Rick was talking about, and tangles, which are predominantly consist of the microtubule binding protein tau. And so, unlike most proteostatic situations, the native state of tau is to be unfolded, and the pathological state is actually to adopt a paired helical filament structure where you have a polymer of tau monomers forming these twisted ribbons of filaments that ultimately form neurofibrillary tangles. These are ubiquitous lesions in normal brain aging, but also can occur in other dementing disorders and brain injury without aging. So just to hammer on tau a little bit more and the aging connection. So aging drives tau pathology irrespective of Alzheimer's disease. So there are 6 stages of tau pathology, actually 7 if you count stage 0. So in stage 0 there is no tau pathology, and that's what you see in young people. By Brock stage 1, you have neurofibrillary tangles in the interadenal cortex, and by the age of 40, between 15 and 20% of individuals are already developing tangles. And this progresses at an approximately linear rate till by the age of 80, essentially everybody has neurofibrillary tangles, whether they're cognitively intact or normal. And typically, onset of Alzheimer's disease is in this stage 3 to stage 4 sort of range. If you look at 80-year-olds, something like 40 to 50% of the population will be, qualifying for Alzheimer's disease based on pathological criteria at that point. So the interesting thing about tau pathology versus amyloid pathology in Alzheimer's disease, and you do have to have both to be diagnosed with Alzheimer's disease, is that, well, plaques are thought to initiate disease progression. They do not track well with cognitive dysfunction. Tau, on the other hand, does. So you can pretty much draw a straight line between cognitive decline and tau pathology. So there's a strong correlation with the degree of tau pathology and the severity of dementia. So that's enough about disease and general aging and tau. I'm going to move on to our model, which is C. elegans. And thankfully, all of the previous speakers I have to thank that have talked about C. elegans, because I'm not going to spend much time here. Essentially, why we use worms is for the experimental advantages. And the chief experimental advantage for us, at least in the context of this talk, is classical forward genetics. So just to describe the model that we've set up, we set about making a model for human tauopathy. That is the tau pathology seen in Alzheimer's disease, normal aging, and a variety of other dementing disorders. So we took the human tau cDNAs and drove them in all C. elegans neurons. And this causes some phenotypes, which we'll discuss in a minute. Also expressed, so shown over here is the staining. The red is neurons, and the green is just a transgenic marker, so we can clearly see animals that are transgenic. And what we see is that when we drive tau expression in all worm neurons, animals carrying normal versions of the human tau have a relatively mild but progressive motor dysfunction and a shortened lifespan, while animals expressing mutant forms of human tau, mutants that cause frontal temporal dementia, not Alzheimer's disease, but frontal temporal dementia, have a more severe progressive motor dysfunction and decreased lifespan. So I'm not going to do a deep dive into the worm nervous system or any of the neurobiology that goes with it, but suffice it to say that when we look at these animals over time, we see initially their nervous system forms normally, and then it falls apart prematurely shortly after the, well, probably right around the end of their reproductive period. And just before death, their nervous system is essentially completely shot. If we look at these tau-containing dying neurons for the tangle pathology that is seen in Alzheimer's disease, we don't see any fibrillar tau. But what we do see is hallmarks of necrotic neurodegeneration. So this DEG1 mutant shows kind of the prototypical onion skin whorl neurodegeneration-like phenotypes in these tau transgenic animals. We see something that's vaguely similar, and these dying neurons are decorated by immunogold with tau antibodies. So these dying neurons contain lots of aggregated tau. Shown here is some biochemistry. We've done a sequential extraction looking at the accumulation of detergent-insoluble tau, which is shown down here. And wild-type tau doesn't aggregate very much, but mutant tau aggregates fairly extensively. And if we graph the amount of insoluble tau over time as compared to motility, we see initially motility, while still compromised, is pretty good on day 1. By day 3, we start to see accumulation of insoluble tau. Motility declines further. By day 5, even more insoluble tau, even less function. So that's very much in keeping with what is seen in disease. So just to sum up the model, we see a progressive behavioral phenotype that is age-dependent. We see age-dependent accumulation of insoluble tau, tau hyperphosphorylation, loss of neurons and degeneration of axons, and a variety of amorphous intercellular tau aggregates. We also see that tau mutations make all of these things worse. What we don't see in the model is fibrillar tau, frank neurofibrillary tangles. This could be because they don't form under any circumstances in animal models in the time scale of days, let alone weeks. They usually take in the order of months or years, so it just may not be physically possible to make a tangle in the course of a worm's life. So there's a bunch of things one can do with models like this. Rick obviously outlined a bunch of them. You test specific hypotheses, screen for drugs, do reverse genetic screens for modifiers, and I'm going to spend the rest of the time talking about a forward genetic screen. And thankfully, Robert and Sean have both talked about cloning genes in C. elegans by a mapping and cloning approach, so I don't have to spend a lot of time there. But the basic approach is we take worms with our tau phenotype that are severely uncoordinated, we mutagenize them, and then search for worms that have restored locomotion. These are potentially mutations that make the animals resistant to tau, and we call these animals SUT mutants, that it stands for suppressor of tauopathy. By undergoing some mapping and genomic rescue, we can identify mutated genes that suppress the tau phenotype. And shown here, I'm going to show you a little movie. So on the left are the tau-unc worms. If you've seen enough worms at this point, I think you can appreciate these guys are not moving normally. They are not paralyzed completely, at least at this day 1 adult age, but they obviously have some serious locomotion problems. So here's the same tau strain with a SUT2 mutation, and you can see it gets around the plate pretty normally. They're not quite wild type, but pretty close. So SUT2 suppresses the behavioral defects we see in tau. So this screen has yielded a number of potentially interesting candidates. I don't like to declare victory until we've got at least 2 independent alleles, so we're still hammering away trying to find more alleles of SUT3, 4, and 5. But SUT1 and 2, we've identified several alleles that suppress tauopathy. And I'm not going to really talk about SUT1. It's a nematode-specific gene. However, SUT2 is conserved across the animal kingdom, and so we're going to focus the rest of our time on SUT2. So just a little more data about SUT2. I showed you that it suppresses the tau pathology. Interestingly, if we overexpress SUT2, it actually makes the relatively severe tau pathology or motility phenotype even worse in this radiolocomotion assay. If I had graphed N2 on this, it would be up at about the 5th or 6th floor here. But these guys are pretty impaired already, and they're essentially completely paralyzed after you overexpress SUT2. Likewise, overexpression of SUT2 in the presence of tau makes the neurodegeneration phenotype much worse. And I should say that SUT2 on its own really has no phenotype, at least no neuronal phenotype. There are some germline phenotypes I'm not going to go into. But so just to summarize the data so far, SUT2 loss of function and SUT2 overexpression have reciprocal phenotypes. So loss of SUT2 suppresses motor dysfunction, overexpression exacerbates the motor dysfunction. Loss of SUT2 reduces aggregation, overexpression increases tau aggregation. Loss of SUT2 protects against neurotoxicity, SUT2 overexpression drives neurotoxicity. Neither loss nor gain of function mutations of SUT2 alter polyglutamine toxicity, suggesting this might be a tau-specific modifier. And in both cases, these animals are neurologically normal. So what is this SUT2? SUT2 is a zinc finger-like protein that is conserved across the animal kingdom. All species seem to have a single copy of SUT2. It's a CCH-type zinc finger protein with 5 zinc fingers at the C-terminus. The zinc chelating residue spacing is absolutely conserved. They also have an N-terminal homology domain that looks something like a Pee-wee domain without the Pee-wee motif. It also has a nuclear export and nuclear localization signals. Although the position of those is not conserved from species to species, even from mouse to human. They're in different positions. So we looked in the literature to find out what SUT2 might do, and when we looked there, this is what we found. There's nothing known about SUT2 when we started all this, so it was a rather daunting task to start from scratch with a new modifier gene conserved across mammals and figure out what's going on. But nonetheless, we thought maybe this is going to tell us something new about tau toxicity. So we started all your basic characterization sorts of things. So we look in worms, we look in humans, we see that in both cases neurons contain SUT2, other cells do as well, although in the brain neurons and ependymal cells are the only cells. In the worm, it's a lot of somatic tissues, gonad, motor neurons, other neurons. But interestingly, in both humans and worms, we saw this nuclear speckle-type staining pattern. So we went to a human cell culture model and did some immunostaining colocalization studies with different things that light up nuclear-like speckles. And one of them, SC35, which is a splicing factor, seemed to co-localize pretty much completely with MSUT2. So MSUT2 appears to be localized in the nucleus to nuclear speckles. So we looked in the brain and in mouse and human and HEC293 cells. There's a variety of SUT2 isoforms matching the predicted messenger RNAs that are produced. When we look both in worms and human cells, we interestingly see that tau overexpressing worms or cells have an upregulation of SUT2. We don't know what that means for sure, but that is certainly something of interest that we're still investigating. So the big question, of course, is what is the normal function of SUT2 and how is this protecting against tau when we get rid of it? So the next thing we did is just went out looking for things that interact with SUT2, hoping that that might tell us something about SUT2 function. So we started with a yeast 2 hybrid screen and identified about 30 cDNAs that interact with worm SUT2 in this 2 hybrid screen specifically. And of those 30 cDNAs, every single one was an isoform of ZyG12. So we had 30 independent isolates that all interact with SUT2. This ZyG12 is a hook protein. We subsequently identified the human homolog of ZyG12, hook 2. There's actually 3 hooks, but only hook 2 binds to the mammalian SUT2 protein. And there is a previous literature on hook 2, thankfully, and it is involved in agrosome formation in cell culture models. So what is an agrosome, you may ask? And there is some controversy about how relevant agrosomes are to human disease, but at least in cultured cells, the agrosome is a structure that forms when the cells have an excess of aggregated proteins. So normally, when the cell encounters an unfolded protein, there are kind of 3 options. One is to degrade that protein, or one is to refold the protein and restore it to its normal function. A second option is to degrade it by the proteasome. And the third option is if you let it sit there for a while, it will aggregate. And then none of these other options are possible. You've got to deal with this aggregate. The aggregate won't fit in the proteasome, and really the only pathway left to get rid of it is to send it through an autophagy-mediated degradation pathway. So the way a cell typically does this, if confronted with too many aggregated proteins, is transport those proteins to a central location near the centrosome, which can then be a substrate for autophagy-mediated degradation via the lysosome. So, with this data in hand, we decided we wanted to build a cellular model for tau-containing agrosome formation. So we took cells and overexpressed the same isoform of human tau that we'd been using in worms. And we noticed that in a few % of cells, we saw something that looked like agrosomes, but it wasn't very satisfying. So we looked for ways that one normally induces agrosome formation, and those are inhibition of the proteasome or inducing oxidative damage, heavy metal treatment, things like this. So with a little bit of proteasome inhibitor 1, we then were able to produce nice agrosomes in most of our cells. We spent some time looking to make sure that these cells really did have agrosomes. So we see that, what is this? This is hook 2. Hook 2 is recruited to agrosomes, has been previously published. It's also true for tau. Dynein becomes localized to agrosomes, has been previously published as well. And we looked at a bunch of phospho-tau epitopes, which are abnormal species of tau that are seen in dementing disorders. So tau is normally not phosphorylated at a very high level under normal circumstances, but in pathological conditions, it becomes highly phosphorylated prior to its aggregation. In our agrosome model, we noticed that while tau is phosphorylated under general overexpression, it becomes specifically recruited to the agrosome upon agrosome induction. And in fact, that's true for a variety of abnormal conformations and phosphoepitopes of tau. I don't have time to show all the data. But for all of these things, they're pretty much normally present in our cell model. When we form agrosomes, they become recruited specifically to agrosomes. So this allowed us an assay to address what the mammalian SUT2 might be doing in these cultured cells. So we knocked down MSUT2 under conditions of agrosome formation, and what we see is that the agrosomes are diminished in number and size and tau content, while overall tau levels remain unchanged. So if we look at this in a biochemical assay, again, this is a sequential extraction of tau. The relevant experiment is down here. When we, in untreated cells, we have an abundance of detergent-insoluble tau. This is aggregated tau. And when we knock down MSUT2, that is dramatically reduced. If we knock it down in cells that have agrosomes formed, we again see a reduction of insoluble tau. So this is all consistent with what we see in worms, where loss of MSUT2 promotes the clearance of aggregated tau. So the party line on how agrosomes are gotten rid of is by autophagy. So we wanted to see what might be going on in autophagy. So we took a reporter system developed by Kraemer et al., where the LC3 protein, a marker for autophagosome formation, has been double labeled at one end with RFP and at the other end with GFP. And so immature or resting LC3 is sitting there being yellow. It's got green and it's got red. And when autophagy is induced, you will see cleavage of LC3 and acidification of the vesicle such that the GFP signal is lost completely from the vesicle and you get a red vesicle it forms. So if we look at resting cells carrying this reporter, we can see that LC3 is this nice greenish yellow over here. And there's very little, although you can occasionally see little tiny spots of low-level autophagy. When we knock down MSUT2, we see a dramatic upregulation of autophagy. This at least tells us that MSUT2 has something to do with autophagic induction, although how it's doing that from the nucleus still remains to be a mystery. If we look at agrosome formation in the context of this reporter, we do see when cells have agrosomes, there is an upregulation of autophagy. But when we knock down MSUT2 in the same context, we see a dramatic increase of autophagosomes that are recruited to the site of the agrosome. So this all seems good. It's consistent from worms to cells. So what's going on in aging-related disease? And we took a look at Alzheimer's disease and characterized MSUT2 a little bit. And so what we've got here is temporal cortex brain lysates from cases, or from controls in cases. These are age-matched and ascending age, so I think the youngest cases are 61 and 62, and these guys are in their 90s. But we don't really see any age-dependent changes that we can discern here, but what we do see is a difference between the levels of MSUT2 in cases and controls. It is dramatically decreased in Alzheimer's disease relative to controls. So we went and looked at postmortem tissue by immunostaining, and we see that MSUT2 is really present in two places in the normal brain. It's present in the lining, or the ependymal cells, and there's no difference between cases and controls in ependymal cells. And it's present in neurons, and there's a dramatic difference between what we see in neurons. So if we look in and around the hippocampus, we can see normal neurons have lots of MSUT2 that are these punctate nuclear speckles. In an AD, there's very little, if any, MSUT2 present in the neuronal nuclei. So there's been some sort of specific loss of MSUT2. So just to summarize, loss of SUT2 prevents tau aggregation and neurotoxicity in our worm model. Overexpression exacerbates this aggregation and toxicity. Human MSUT2, we know, binds to HOOK2, a component of tau agrosomes, and knockdown of MSUT2 clears insoluble tau. We also know that knockdown promotes autophagy activation, and this somehow promotes clearance of insoluble tau. We know that MSUT2 is also decreased in Alzheimer's disease, and its levels are negatively correlated with aggregated tau. So I'd like to thank the people that have done most of this work, and our collaborators, and funding agencies, and you all for listening. Thanks.
We have time for a couple of questions. Are you
That's correct.
Could this mean that simply the aggregates formed by tau in this model are just smaller than they are in humans?
Could be. I mean, everything's smaller, right? And the neurons in C. elegans are absolutely tiny compared to what you see in mammals. So they may be, but I mean, even we looked by EM, we looked hard, and we didn't see anything that resembled fibrillar tau. So I'm not saying they're not there, but we couldn't see anything that looked fibrillar.
When you say that deletion of M suit clears the tau aggregates, does it mean that you by which method do you observe this? By fluorescence?
No, by biochemistry. By biochemistry. So it doesn't change any of the abnormal conformation dependent antibodies, which are pre-aggregate conformations. It doesn't change phosphoepitopes. So there is no imaging method that works. If you put tags on tau to do FRAP or things like this, it changes the nature and toxicity of the aggregates quite a bit. So I don't think there are really any good light microscopy methods for imaging tau aggregation in this model, unlike with poly-Q.
Yes, I have a question. Actually, it's not my question. I'm talking to translate. So the question is, if and how acetylcholine esterase inhibitors affect mobility?
So cholinesterase inhibitors, these guys are more resistant to cholinesterase inhibitors, which is generally thought to be an indicator of compromised neurotransmission. But in terms of a therapeutic, it's a very, very modest sort of improvement, I would say.
There is something that I do not understand. It's elegans, you told us that SUB2 loss is good. The homolog of SUB2 is 10 SUB2, and the in Alzheimer's disease is decreased. Should be the contrary.
So, okay.
How do you explain this?
So we're going to take a little trip to my fantasy world here. So this may not be the way it is, but this is what I would like to think. Might be true. So if we've shown that loss of SUT2 makes cells, worms, resistant to tau toxicity, and overexpression makes them sensitive. In Alzheimer's disease, I predict that what's going on is that neurons with high levels of SUT2 are killed by the tau pathology, while neurons with low levels of SUT2 are resistant. And so when you look in the AD brain, all that's left are neurons with low levels of SUT2. There's some other circumstantial evidence in support of that, in that if you look in brain regions that are resistant to tau toxicity normally, like the cerebellum, there is no SUT2 there. So it's all consistent, but with postmortem studies, one never knows causation, of course.